Hello and welcome to this Revision Monkey video on what's going to be in the Combined Biology Paper 1 exam for 2023, and this is for Foundation Tier for the AQA specification. So what should we expect in this 2023 exam? Well, in Biology Paper 1, about 10% of it will be calculations, about 15% will be required practicals, and then you'll also have lots of tables, lots of explain questions, compare questions, lots of graphs, and evaluate questions as well. So let's look at how to answer an evaluate question. Now, not only can this come up in Biology Paper 1, it can come up in any of your 6 exam papers for science. So evaluate, let's have a look at a typical question. It says evaluate the use of embryonic stem cells and adult stem cells to treat paralysis. So what that word evaluate actually means, but they don't tell you, is give advantages and disadvantages of both embryonic and adult stem cells to treat paralysis, and then give your own conclusion as to which one is best and why. They don't write it like that because it's too wordy, so instead they use the word evaluate. But you must know, to get marks in these questions, you must give advantages and disadvantages of whatever's in the question, and then come to your own conclusion about which one is better. And there's no right or wrong for that, okay? You just take one particular stance point and suggest which is better and give a reason why. For example, if we were looking at this question, you could have sentences structured like this. An advantage of an embryonic stem cell is, another advantages of an embryonic stem cells is, and you're putting it on a plate for the examiner. You're really showing them that you're listing those advantages. And then disadvantages as well, making sure you're putting it on the plate and listing your disadvantages. And then you have to do the same for the other one. So, for example, adult stem cell, again, make it really clear that you're saying whether it's an advantage or a disadvantage. And then finally, you write your conclusion. So good to start with the word overall, and then also have the word although in there, because that gives it a justified conclusion. So, for example, for this stance point, you could put overall, I think adult stem cells are better because although they have a limited ability to differentiate, I think they should be chosen because they are less likely to be rejected by the patient. So you must justify why you're choosing your one you're choosing at the end. Don't just say, overall, I think you should choose adult stem cells. You need to justify your answer. So if you see that word evaluate, don't forget advantages, disadvantages, and a justified conclusion. That's what it means. Compare questions, again, they just write compare, but they don't tell you what to do. So you need to remember, if you see a compare question, for example, compare a bacterial cell to an animal cell, what it really wants you to do is give similarities and differences between bacterial cells and animal cells. It won't tell you that, but you need to remember to say things that are similar and also the differences. But they just give you that one word, compare. So just bear that in mind. For example, you could structure it like this, where you would say similarities, and you would list your similarities, and then you state your differences as well. And you need to get both of these things in the question. Now for the differences, I've highlighted the ending ER, okay, so these make the words comparative. So animal cells are larger than bacterial cells. Don't just write animal cells are large, because that's not comparing it to the bacterial cells. So as many of these ER words, larger, taller, heavier, etc., that you can get in, the better. If you can't use an ER word, use the word whereas. So for example, animal cells have a nucleus, whereas bacterial cells do not. So remember, compare needs similarities and differences in your answer. And let's look at the explain questions. Explain the results in the table. So the results in the table are showing the distance of the pondweed from the light and the number of bubbles per minute. So a little snapshot of data from one of your required practicals. So explain doesn't mean that you write as the distance between the pondweed and the light increased, the number of bubbles per minute decreased. That's writing a conclusion or describing the results in the table. Explain means why have those results happened, and basically just try and chuck in as much science as possible. So some type of things that you could write down would be things like the light intensity is a limiting factor affecting photosynthesis. The closer the light is to the pondweed, the higher the light intensity the plant is receiving. The rate of photosynthesis increases as light intensity increases, up to a point when light intensity is no longer a limiting factor. So you can bullet point all of these key scientific details which explain why those results are what they are, rather than describing the results. And you might increase your explanation further by writing about the chlorophyll is absorbing the light, and one of the products of photosynthesis is oxygen, and oxygen bubbles are released by the pondweed when it's photosynthesizing. Hence, the closer the pondweed, the higher the number of bubbles released per minute. So explain questions are often quite a few marks, so bullet point lots of scientific facts which explain and say why the results are what they are. I just want to highlight now some calculations, some that you might want to brain dump in the exam. And by brain dump, I mean as soon as the examiner lets you begin, if you think these are going to fall out of your brain, you can write them in any white space that you can find on your question booklet. So the equations that you might need to recall for this paper are the magnification equation. Magnification equals image over real. So you could literally write down m equals I divided by r. Okay, as soon as you get in the exam, just in case you need it. Now for foundation, they do sometimes give you the equation, but in case they don't, it's there and you've written it down ready in case you need to use it. Another one is magnification equals the eyepiece times lens. So this one helps you work out what magnification on a microscope you're using. Percentage change is a really important one for paper 1. And to calculate percentage change, you do the change divided by the original and then multiplied by 100. And then finally, this could come up on any of your 6 papers, is uncertainty. To calculate uncertainty, you do the range divided by 2. So just 4 important equations I recommend you learning for this exam. Here are the required practicals for biology paper 1. So there's quite a few. There's photosynthesis, enzymes, microscopes, osmosis and food tests. And some of these will come up and 15% of the marks will be on required practicals. So microscopes, first of all, are something that we can use to look at very small objects like cells. So we might prepare our slide here with our cells on. And unless we stain them, they're really difficult to see. So you might want to put a few drops of iodine on them or something like that just to stain the cells. Then a cover slip on top and it gets put onto the stage. We look at the cells using the eyepiece, which is the top bit. This often has a number on it, for example, times by 10. And then we have the lenses below, again, which will have numbers on them, perhaps 4, 10 and 40, for example. And some focusing knobs or focusing wheels. You'll often have a coarse one and a fine one, which I'll just draw on here as well. So the main points then is that we need to start at the lowest magnification. We calculate that by doing the objective lens multiplied by the eyepiece. So for example, if this lens is 4, the eyepiece is 10, we know we're looking at the cells at 40 times magnified. And this one would be 10 times 10, so 140 times 10. So 400. So we start on the lowest one, and to focus these cells, we need to use the focusing knobs. Now, in the exam, if they give you a picture of cells that are a bit blurred, then it's these focusing knobs here that you need to use. To view the cells in more detail, you need to increase the magnification. So if the question asks about how you'd look at more details inside the cell, you need to talk about changing the objective lens. And if you want an even more detail, you might want to use a higher resolution microscope. For example, your electron microscope. This one is just a light microscope. Moving on to the next section of this microscope then is this equation which you're going to need to remember for your exam. Magnification equals image size over real size. Now, they could ask you to work out the magnification, or they could ask you to work out the real size. The image size is what you actually measure. So let's take this question. What I suggest you do as soon as you get into the exam, brain dump this. When you're asked to begin your exam, brain dump it on a bit of white space if you can't remember it. Magnification equals image over real. The pollen grain in the image is magnified 800 times, so that's our magnification. Calculate the real diameter and give your answer in micrometers. Now, if you could do the first couple of steps, that's really good. That will get you 2 out of 3 marks. And if you can do the conversion at the end, that is a bonus. So if we are to measure the image size, we need to get our ruler and we need to measure the diameter of the cell. In this case, it's 4 centimeters. I strongly recommend you measuring in millimeters, as it will be more accurate and it helps you with a conversion later on. So 40 millimeters is our image size. And we've got 800 as our magnification. So we need to rearrange our equation so we get real size equals image divided by magnification, and then we put our numbers in. So real size equals 40 divided by 800. But because we then have to convert it to micrometers, this would give our answer in millimeters. We need to then convert it into micrometers by timesing by 1,000, and that gives us our answer of 50 micrometers. If you do the first couple of steps and work out this as 0.05, then that will get you a couple of marks, but just don't forget these prefixes, nano, micro, milli, etc., to convert to if you need to. Osmosis then, we'll talk a little bit about the content and then the required practical. So osmosis, this is the definition that confuses people. Movement of water from a dilute to a concentrated solution. This means from where there's lots of water, so a high concentration of water, to where there's not so much water, a low concentration of water. So you can use either definition in the exam, but they may well talk about it moving from a dilute to a concentrated solution. So in this practical, we put something in a sugar solution. So outside here, we've got our sugar solution. Now I say something on purpose. Don't be thrown out if what they put in is not a potato. It could well be, but it could be a carrot or a beetroot, or it could be a sweet or anything that contains sugar inside. Because the potato itself or the vegetable or whatever we're looking at has a particular sugar solution inside. So when we put it in the beaker here, in this situation, because osmosis is the movement of water from a dilute to a concentrated solution, the water would move inwards, increasing the mass of the potato. So it's just an arrow up to show increasing the mass. However, in this situation, the solution inside the vegetable is more dilute than the outside. So the water will move out of the vegetable into solution. And in this case, it will decrease in mass. Now, at some point, you're going to get a situation where there'll be no mass change when the solution inside has the same concentration as the solution outside. And we'll look at that in just a minute. Now, the osmotic balance of cells is really important. It's really important that you don't have too much water or too little water. If you have too much water in your bloodstream, your cells are going to take in that water and swell, And if you have too little water and you're dehydrated, your cells are going to shrivel up and will not function very well. That's why when you have, after exercise, you'll often drink an isotonic drink. That has the same concentration as sugar as your blood. So when you're drinking that, it will quickly replenish the sugars that have been used up in respiration and things like that during your exercise, and it won't affect the osmotic balance of the cells. So drinking isotonic drinks afterwards are really good after exercise. So what results would we get from this practical then? Well, we put our vegetables in different sugar solutions, like so. We'd want to try and cut them so that they were similar lengths and widths. Okay, that's going to be a control variable. And we want to keep them at the same temperature. We're going to measure the mass change, and we're going to change the concentration of sugar solution. So our results may look a little something like this. We've got our different sugar solutions, our mass before. Note that they're slightly different because you can't cut a piece of vegetable or a sweet or whatever it is to be exactly the same mass. So we have the mass of the potato afterwards, and we can calculate the mass change. Now, what you're going to need to do as one of your math skills, especially in biology, is calculate percentage change. So the difference in the mass of the potato would be 3, so the change would be 3. 15 minus 12 gives us 3. And then we divide that by the original, which in this case it started off as 12. So 3 divided by 12 and multiplied by 100 will give us a percentage change for this first one of plus 25%, plus because it's increased in mass. If you do the same for the next one, change divided by original times 100, you get plus 18%. And then the final one, you will get a minus 25% because this one has actually decreased in mass. You might then need to plot these on a graph. So if you're looking at the sugar concentration against percentage change in mass, when it was 0, that was plus 25. When it was 0.5, that was 18. And when it was 1, that was minus 25, somewhere down here. And then you'd be looking at drawing a line of best fit. Now, if this graph was any good, which it's not, they'll often be in a straighter line than I've done there. But we'll draw a line of best fit through using a ruler. And this is the key point. Where it crosses the line, this sugar concentration here, if we were to read it off our axes, they'll give you better axes in the exam. This would be the concentration of sugar solution inside the potato. Because at this point, there's no mass change. So this is the concentration of sugar solution inside the potato. Boiling vegetables, if you were to boil vegetables, this disrupts the cell membranes, so they are no longer partially permeable. So if you put a boiled piece of vegetable inside a solution, you won't get a mass change because those partially permeable membranes are not functioning anymore. Now, typical questions for osmosis. So it could be in relation to the movement of water inside and outside of guard cells, for example. Guard cells are the cells that open when they are turgid and full of water, and they close when they are flaccid. You could talk about the movement of ions and water in relation to guard cells. They may talk about osmosis in relation to root hair cells, which we'll talk about later on, or just general movement of water in body cells. Moving on then, food tests. So the food test for fats is not one that you're going to be asked to recall anymore. So you need to concentrate on starch, sugar, and protein. So first of all, you'd crush up some food, mix it with some water, add it to a test tube so you've got your food solution in here. And then there's 3 tests that you're going to have to remember. The first one is the test for starch. This is iodine. You add iodine, and if starch is present, it will turn from orange to black. The next one is sugar. That's Benedict's solution, which will change from blue to orange. Now you may see other words. If there's a little bit of sugar, it may change green. If there's loads, it may change a red colour, but blue to orange is your main colour change. This one requires a water bath at around 75 degrees. Okay, so for the sugar one, you're going to need to heat it up in a water bath. And finally, protein is Bio-Ret test, which turns from blue to purple. Moving on to enzymes. We're going to do the enzymes required practical, so I'm just going to take this moment to do a bit of the theory behind enzymes. They are made out of proteins. Biology paper 2, you talk about DNA coding for amino acids that make proteins, and proteins fold up into a very particular shape. This here, hopefully you'll be able to recognise as the active site. And the thing that goes into the active site we call the substrate. So one theory is the lock and key theory, which says that the substrate is an identical match to the active site. And a slightly more complicated theory is called the induced fit theory. And that just says when the substrate goes in, the active site just changes shape ever so slightly to grip the substrate in. But nevertheless, you're going to have a specific shape that is going to match the active site. If the enzymes are at too high a temperature, they denature. You cannot say they die because they are not a living thing, they're a protein. And what that means is the active site changes shape and that will no longer bind the substrate. Okay, so when they denature, the active site actually will change shape. And if the substrate can't bind, then it can't go in and be split, for example, from a large molecule into smaller molecules. Let's look at the practical then. This practical focuses on amylase. Amylase is an enzyme that breaks down starch into sugar. So we start off by putting a certain pH in, because this time we're going to look at how pH affects enzyme activity. So the independent variable we're using is pH. We could use a range of buffers like pH 3, pH 5, pH 8, etc. We're going to measure the rate of enzyme activity, and we're going to control temperature and the volume of enzyme or starch that you're using, etc. So we're going to put the pH buffer in there, and then we're going to add the amylase on top. And we're going to heat it to 37 degrees C. We're going to try and control that because that is the optimum temperature for enzymes. We're going to set up over here a spotting tile by adding iodine into every single well, and that will go an orangey brown colour. And then we're ready to add our starch. So the final thing that we add into our test tube is our starch. And then straight away, we will take a sample out of our test tube and we will place that in the spotting tile. So we'll take a sample out and we will then be able to put that in our spotting tile, and our iodine is going to be testing for the starch. So if starch is present, that iodine is going to turn black. Can't change the colour at the moment, so I can't turn it black, but these ones here will change black. And again, I would take more of my sample out, do it in the next well, and again, that would turn black. All the time that the starch is still present within here, our samples are going to go black. But eventually, the amylase will have broken down all of the starch into sugar. So at one point, you're going to put your sample on here, and it is going to remain orange. And at that point there, you know that the amylase has broken down all of the starch. So you do it every 30 seconds, 30, 60, 90, 120, etc. And let's say we got to this point and all of these were black and all of that stayed orange. We would know that the reaction had finished and the enzyme had finished breaking down the starch. Now, to calculate a rate from that, if you're doing foundation, I wouldn't worry too much about this, but higher tier, to calculate a rate from this experiment, we need to know the equation 1000 divided by time. Final one that I'm going to briefly go through is photosynthesis. Now, these are fully in detail on another video, if you want a bit more detail. So the independent variable is the distance between the lamp and pondweed. That, in turn, changes the light intensity. The dependent variable is the number of bubbles per minute. In this case, you need to be talking about either this or in terms of a rate of photosynthesis. You could have, if you wanted to, and you may see in the exam, a gas syringe attached to it. And rather than measuring the number of bubbles per minute, you may well measure the volume of gas produced. The control variables then, temperature, power of the lamp, so you might want to stick with, for example, a 40-watt lamp, the length of pondweed and the time of pondweed, and you start at a certain distance, count the number of bubbles per minute or the volume of gas produced per minute, and then you would do that at a closer distance as you move the lamp, and so on and so forth. Next, I'm going to look at some maths skills. First one has a lot of relevance to biology paper 1, and this is surface area to volume ratio. Let's work it out for this one. So if we're looking at the surface area to volume ratio, we need to calculate the surface area. So let's remind ourselves that we calculate the area of one side and then multiply it by 6 because there are 6 sides. So this one would be 1 times 1 and then multiplied by 6, and that would give us a surface area of 6, and the volume, length times width times depth, so 1 times 1 times 1 will give us a volume of 1 centimetres cubed, and obviously that was centimetres squared. So that would give us a ratio of surface area of 6 to 1 volume. And you may need to do this in biology or later on in chemistry or physics, so it's a skill that we need to know. For this one, surface area would be 3 times 3 for the area of one side, multiplied by 6, which will give us 54 centimetres cubed, squared, and the volume 3 times 3 times 3, which is 27 centimetres cubed. If we put those as a ratio, we'd have 54 to 27. But they're quite difficult to look at and compare at this point. So because this one's at a 1, if we try and also get this side to a 1 by dividing it by 27, that will give us 1. And we need to do the same to the other side, divide by 27, and that gives us a ratio of 2. So you might need to do that calculation and also understand that smaller organisms, if we suggested that this was an animal, for example, a smaller animal has a larger surface area to volume ratio than a larger animal. So something like a bacteria, oxygen can diffuse really easily into the bacteria because it's got a large surface area to volume ratio. However, for us, it's a lot more difficult. That's why we have things inside our body to increase surface area, like our alveoli and like the fish, they have their fish gills and things, which we'll talk about in detail in a minute, and villi in the small intestine. We have things to try and increase our surface area. So a bit more maths, and then I'll come back to those exchange surfaces. Averages, you might be asked to actually write down like this how to calculate an average, so make sure you know how to do that. If we look at the first trial, we carefully check for anomalies, and they all look quite similar to each other. So we would add all of those up and show you're working out in case you make a mistake. Add all of those up and then divide by 5, and that gives us our answer of 11. For set B, you need to look carefully and you'll notice there's an anomaly here. So just circle that one and leave that out of your calculation. 9 plus 9 plus 10 plus 11 divided by 4, and that will give you your answer of 9.75. However, next learning point, you must round this to the same number of decimal places collected in the table. So they might talk about that as being a mistake if someone's done it wrong. So round that up to 10. Uncertainty, if you're writing that list of calculations to remember, this is definitely one. Uncertainty is range divided by 2. We don't know where it's going to come up. It could come up in biology, it could come up in chemistry or physics. So uncertainty is the range. So if we looked at uncertainty of result A, the range is 13 to 10. So 13 minus 10 will give us 3. And then we divide by 2. So our uncertainty of those results is what we always put in front, plus or minus 1.5. They may ask you to calculate uncertainties and draw them on a bar chart, for example. There's your bar chart, and you might have calculated your uncertainty as plus or minus 1.5, and the reason why it's plus or minus is you would show it plus 1.5, minus 1.5, for example. And the bigger the error bars, the more uncertainty you have. Median then. Next, write the numbers in ascending order. The median is the middle number. So if we took these results here, we would write them in ascending order: 19, 22, 22, 25, 26, 27, and 28. So make sure you can write it out like this and also then choose the median number correctly. And the mode is the one that appears most often. So this case 22 appears twice. So that is our mode. Graphs, nice and quickly on graphs then. Not too much to say here, only that you need to show you're working on graphs. So if you're asked to work out something from a graph with a ruler, go as accurately as you can to the graph and across, showing that working on the graph and then writing your answer. If you have a relationship like this, for example, don't forget that you can draw a curved line of best fit. Now this one is sitting outside of the curve, so I treat that as an anomaly and I carefully draw my curve as close to as many of the points as possible. Don't join them up. Similarly, you could have something that looks like that. No straight line necessary because you can see that it's got a curved relationship. So carefully put a curve on there. A directly proportional one is a straight line through the origin. So it could be like that, or like that, or like that. They're all directly proportional if it's a straight line. And inversely proportional means that as x gets bigger, y gets smaller. So it will look something like that. So as one gets bigger, the other one gets smaller. Tables then. So we've got sketch graphs. You might be able to draw a sketch graph of the data. Some people are lazy and lose easy marks. Take the whole title from the table. Don't make something up or convert it or change it in any way. Take that whole thing and write it underneath the sketch graph. Take this whole label, write it on your sketch graph. And then you would have a look at the relationship. So it goes up, and then look here, it flattens out. So carefully look at the data. If you're asked to describe this, you need to talk about it increasing and then leveling off and staying the same. So your sketch graph would look something like that. Labels on and the sketch curve. Right, let's go through a little bit of content now then. First of all, I'm not going to go through these in detail. Hopefully you know cells by now. You need to name all the parts and all the functions. I'm going to highlight a couple of points though. Simple cells without a nucleus and mitochondria, without those subcellular structured parts, are prokaryotic or prokaryotes. And ones with subcellular structures that are structured, like mitochondria and chloroplasts and things like that, are eukaryotic cells. Okay, the difference between plant and animals is that plants have the cell wall, the chloroplast, and the vacuole. They have all of the other things that the animal cell does, if you're asked to compare them. And the bacterial cell often has a loose bit of DNA and also sometimes has a ring of DNA as well called a plasmid. Not all of them, but some of them have a flagellum for moving. The cell cycle then. In this topic, we're talking about mitosis, okay, for growth and repair. From the moment that you are a bundle of cells as an embryo until, well, you're still doing mitosis now, okay? You're doing mitosis now for growth. Okay, you're still not fully grown, and you're doing it for repair. So, any cell division that starts off with one cell and ends up with two identical cells, we're talking about mitosis, not meiosis. Meiosis is paper 2, so get that out of your minds and think just about mitosis. So you might see this kind of pie chart along with it. This shows the time that it takes for each stage of this cell cycle process, because actually the mitosis part of it is a small amount of time. So we've got growth and DNA replication, because if we start off with one cell, like we saw before, that's got things like cytoplasm inside, ribosomes, mitochondria, and all of that needs to double up so that we can make 2 cells at the end of this process. So that takes a long time. Also, the nucleus contains 46 chromosomes, and we need to double that up as well. And when they do that, they make an identical copy of themselves, so the chromosomes will look a little bit like X's, like that. So they've copied themselves up, ready to be split into 2 cells. Now the mitosis bit, okay, that's all the growth and replication. The mitosis bit is the splitting of the chromosomes to make 2 different nuclei. So the chromosomes, will line up in the middle, and then they will be pulled apart. And you'll start to form 2 new cells. It might have a bit of a, let me draw it over here, might have a bit of a weird shape like that as it starts to form the 2 cells. So it's now got 2 different nuclei in, each have identical 46 chromosomes, and then this is the mitosis part, okay? So this part here, where the chromosomes are splitting apart, and where the 2 new nuclei are formed, that's mitosis. And the final bit, they might split up this bit into saying the final bit here is when the cytoplasm divides and 2 new cell membranes form. So when they actually go from that bit to 2 new cells, that's the final bit of the cell cycle. Stem cells then, there's a couple of places. Think about where in the body and the other place that you can find stem cells. So stem cells are found in bone marrow. Those are quite good. They can differentiate into things like blood cells. However, the stem cells that are really good are embryonic stem cells, because you started from an embryo, so they make every single cell type. They can turn them on to make a muscle cell, a liver cell, cells to replace your cornea in your eye, whatever it is. They can turn into any type of cell. Now, the trouble is with stem cells is they don't contain your DNA if they're taken from a random embryo, and perhaps that will be rejected by your body. So this is the next stage up, really, that scientists and doctors are looking at the moment, and that's therapeutic cloning. That's when you take an egg cell donated by a woman with her DNA in it, and you take her DNA out, so you have an empty eggshell. You take one of your body cells with your DNA in it, and you remove that and combine it with the empty egg cell. So you've got an egg cell that's capable of dividing, and it's got your DNA in it. So then you'll stimulate that to divide. Sometimes that's by an electric shock. You'll stimulate that to divide, and it will start dividing by mitosis to produce embryonic stem cells. Now, if you were to leave that in the right conditions and implant it into a woman, that would make a clone of you, okay? But they don't get to that stage. They only get to the stage where they're making cells. And then you can use that to make muscle cells, liver cells, etc. Okay, let's move on to transporting particles. And I'll try and link these into other themes of the topic. This is an important theme that runs through Biology Paper 1. The first one that we'll look at is diffusion, because we've already looked at osmosis in a bit more detail. Diffusion, over here definition, movement of particles from a high to low concentration. So in both of these cases, concentration is higher on the outside compared with the inside. So oxygen would diffuse into the cells. However, this is the bit that's important. The rate of diffusion depends on the concentration gradient. That is the difference in the concentration between inside and outside of the cells. A big difference like this one, you're going to get a high rate of diffusion. A small difference like this one, the rate of diffusion is going to be slower. Still going to move in the cell, but it's slower. So a key point, The bigger the concentration gradient, the faster the rate of diffusion. Comes up several themes throughout the paper. And one thing we'll talk about is exchange surfaces. So I mentioned them briefly before. Because we've got a small surface area to volume ratio, we're going to need things like the alveoli in our lungs with a large surface area. So we take in oxygen and that needs to move into our blood. If our blood wasn't flowing, we wouldn't be very good at exchanging oxygen into the blood, because we need to maintain a difference in concentration. We need to maintain a concentration gradient so the blood takes away the oxygen as it flows. If you didn't have that, then you wouldn't be able to diffuse the oxygen over. Similar way to fish gills. So fish gills, fish need to take in oxygen, and they have gills with filaments, again, to increase the surface area. So these are the filaments. And they have even more surface area on top by these little fins called lamellae on top. Whole purpose is to increase the surface area. Now, through those gill filaments, you have water going in one direction and blood going in the opposite direction, because the water is going to is carrying the oxygen that will diffuse into the blood, and you need the blood to take it away in the opposite direction to maintain that concentration gradient. Okay, the bigger the difference in concentration, the faster the rate of diffusion. You've also got the villi in the small intestine, which are the folds to increase the surface area. So, osmosis and active transport, but this time within the context of a root hair cell. Water will move in by osmosis from a dilute solution to a concentrated solution, or you can say from a high concentration of water to a low concentration of water. And ions, think about how they're going to move. Look, these ions in red are in a low concentration on the outside and a high concentration on the inside. So those will move in by active transport. This is a kind of a linking thing that they might ask in a longer answer question. Active transport goes against the concentration gradient. Against the concentration gradient. Okay, because you're going from where there's fewer particles per volume to where there's more particles per volume. So this is the normal concentration gradient here, conc gradient. But active transport goes against that. Okay, so talk about it going against the concentration gradient. And for this, it needs energy. And in the cell, the part of the cell that provides the energy is the mitochondria through respiration. So talking about 6 markers here, how things move into the cells, and the fact that you've got mitochondria that provide the energy for active transport. Then the water and ions that have moved in, we will then see those travel up through the xylem. So water and the ions move in an upwards direction only through the xylem, and we call this the constant transpiration stream. The xylem itself is dead. It's strengthened by lignin, because if you just had dead cells, it would be very brittle. And it's hollow to allow the water and the ions to move up. And the loss of water from a plant is called transpiration. So if you have a leaf, we'll talk about the leaf later, you've got holes on the bottom, and unfortunately, water can be lost. And the factors that increase it, if you increase the temperature, that will increase the water loss or the rate of transpiration. If you increase the light intensity, because it will be doing more photosynthesis, all the stomata will be open. And think carefully in terms of concentration gradients. If you decrease the humidity, that means there'll be less water in the air. Okay, humidity is all about the water in the air. If you're low humidity, you're going to have a bigger difference between the concentration of water in the leaf to outside. So you're going to have lots of water leaving. And if you increase the airflow, a little bit like the blood in the fish taking away the oxygen, increased airflow would take away the particles from underneath the leaf, again maintaining the concentration gradient. The leaf, so I'm trying to link this with photosynthesis, again trying to link your ideas with 6 markers and things like that. So can we name all the tissues? We've got to link it to this equation. So carbon dioxide and water make glucose and oxygen. So the epidermal tissue is the top layer that covers the leaf. The waxy cuticle is right on top of this, and now this is to reduce the water loss. Okay, it's not anything about keeping it waterproof or anything like that. It's to stop the water coming out of the leaf into the atmosphere. The palisade cells are part of the mesophyll layer. Okay, this whole layer in the middle is the mesophyll. It's separated by the palisade mesophyll at the top. Specialized cells full of chloroplasts for photosynthesis. Spongy mesophyll at the bottom, gaps in between to allow carbon dioxide to diffuse up to the palisade cells, and again covering epidermal tissue at the bottom. You've got guard cells, which are here and here. These control the opening and closing of the stomata. You might just see the word stoma or stomata, stoma being one, stomata being many. And the final vessels, xylem and phloem. So if you're linking this to photosynthesis, you need to be talking about carbon dioxide diffusing through the stomata, which are controlled by the guard cells, the spongy mesophyll having gaps to allow the carbon dioxide through to the palisade layer, and this having specialized cells full of chloroplasts for photosynthesis. The epidermal tissue is thin to allow sunlight through, and the xylem is what's going to bring the water for photosynthesis. Now, in that process, you are going to need to know about limiting factors that affect the rate of photosynthesis. So, light intensity. If you increase light intensity, you will increase the rate of photosynthesis. But the point that it levels out here, at this point, light intensity is no longer a limiting factor. Okay, if it was down here, it is a limiting factor, because if you increase the light intensity, you'd get more photosynthesis out of the plant. Carbon dioxide, luckily, has the same pattern, so it's nice and easy to remember. And the temperature one, because it's related to enzymes, has a little bit of a different look. Because if you increase the temperature, the rate of photosynthesis increases up until its optimal point, and then the enzymes will start to denature, decreasing the rate. The glucose that we make in photosynthesis then, again trying to link together some ideas about the leaf and the phloem and photosynthesis. The glucose that we make, or not that we make, but plants make, needs to be transported. And that is transported in the phloem. And it's used for several things. Could be a 5 marker. For respiration, so the opposite of this is respiration. You could oxidize it straight away with oxygen to release energy. It's stored as lipids, so fats and oils in things like seeds. Stored as starch. For example, a potato is a big starch store. It's used to make cellulose for cell walls and to make amino acids, which make proteins. Now, this one uses nitrates from the soil as well. So the bottom one, the plant needs the glucose and nitrates which it gets from the soil. Then it will travel in the phloem. These are the living cells, similar to xylem. In fact, they're tubes that transport things, but they've got pores between the cells. They're not completely hollow. And sugar can be transported in both directions. So phloem transports the food, and the food is obviously sugar or glucose. And we call this process of moving sugars around translocation. Linking on to respiration then, there's aerobic respiration and anaerobic respiration that we need to be aware of. So definitely being able to recall these things is key. Again, if it's something you struggle on, then this might be the thing that you brain dump when you start the exam. So glucose reacting with oxygen makes carbon dioxide and water. We show that by the simple equation. C6H12O6 is our glucose, reacting with oxygen, making carbon dioxide and water. And it's not written in the equation, but this is obviously, the purpose of this is to release energy. That's what respiration is, the release of energy from glucose in your cells. And as a reminder, happens in the mitochondria, so it happens in animals and plant cells. Anaerobic respiration in animals, so that includes your muscles, turns glucose straight into lactic acid. If there's not enough oxygen, you get a lactic acid buildup and that causes cramps. And in plants and yeast, the anaerobic respiration equation is different. Glucose is converted to ethanol and carbon dioxide. This product here that's made, is used in alcoholic drinks. And this product is used in the baking industry to help the bread rise. Now, when you're making bread, you actually make ethanol as well, but as soon as you put it in the oven, that boils and is taken away. Okay, let's move on to the heart and lungs then. So with the heart, you need to name the major vessels going in and out of the heart. So you've got the aorta, which is the main artery. This part of the heart is the left-hand side, and this is the right, so it's the opposite to what you think. This has a very thick muscular wall on the left-hand side compared to the right, because the aorta takes the blood around the whole body. So it needs a thick muscle to push and force the blood out a longer distance. When the blood goes around your body, oxygen will diffuse into your cells and your blood plasma will receive the carbon dioxide, and that will come back in through your vena cava, which is your main vein. Now, if you remember those two, the other ones are easy because on each side of the heart, you have one artery and one vein. Okay, so if that helps you remember which way the pulmonary one goes, that might help. But if you've got the blood coming back from your body, it needs to go away to the lungs. So you need to send it through the pulmonary artery. It goes to the lungs, which we'll talk about in a minute. It gets its oxygen through diffusion through the alveoli, and it comes back in through the pulmonary vein. You've got cells in your heart that control your heartbeat. They are in the right atrium. The top chamber is the atrium, and these are the pacemaker cells. And you've got valves in between the atrium and the ventricle, which is the bottom section, atrium, ventricle, valves that control the blood flow. And those are the things that can go wrong and you might need replaced. Up to the lungs then, you've got the trachea, you've got the bronchus, and then splitting off for a larger surface area, the alveoli. The three blood vessels, the artery, the vein, and the capillary. The artery is the one with the thick muscular walls, so it have a relatively large lumen, but quite thick muscular and elastic walls because blood flows at high pressure. The vein has a very large lumen, which is the opening, and it has a valve inside to stop the backflow. And the capillary are really thin, just one cell thick, because out of there, all the oxygen needs to diffuse to the cells around it and sugar and things like that. Okay, exercise. I'm going to highlight to you the keywords that are going to come up in exercise questions. Again, if you're looking for longer answer questions especially, they're going to be looking for an increased heart rate, an increased breathing rate, and maybe breathing volume as well you can put. Because you want increased blood flow to the muscles, because you need more oxygen for respiration to release more energy. So in these kind of exercise questions, as long as you're getting the words energy, respiration, oxygen, blood flow, you're going to be hitting the marks and just making sure you're saying whether they're increasing or decreasing. Okay, we're trying to do a bit on digestion and enzymes. So when you digest food, it goes through your mouth first of all, and it goes through your esophagus into your stomach. Your stomach has an acid in it called hydrochloric acid. That is one of your defenses against disease. When you eat food, if you take bacteria in, that acid will kill the bacteria. You've then got the food going through the small intestine where nutrients are absorbed, and then the large intestine where water is absorbed. Now, the conditions in the stomach are acidic. However, the conditions in the small intestine are alkali. So enzymes that work in the small intestine love alkali conditions. Enzymes that work in the stomach love acidic conditions. So what we need to do is stop the food from taking all of that acid into the small intestine and ruining the conditions for enzymes. So the final thing is we've got bile, which is produced in the liver and stored in the gallbladder, which is next to the liver. And it neutralizes stomach acid, and that's one thing that it does. And it also emulsifies fats, which means it takes the big pieces of fat and turns them into smaller particles like so, increasing the surface area so the enzymes can get to it. Let's add some enzymes on then to our diagram. I thought about this today, so I'm always thinking about a way to link things up to learn them for you. If you remember it as apple, APL, apple, you'll remember it in the order that they appear in the human body. A is amylase, that first starts working in the mouth, and amylase breaks down starch into sugar. P is protease, and that first starts working in the stomach, and protease turns proteins into amino acids. And finally, lipase starts working in the small intestine. And in fact, all of them work here. So all three enzymes work in the small intestine. That's where most of the nutrients are digested and stored. And lipase turns lipids or fats into fatty acids and glycerol. Okay, so all three enzymes work in the small intestine. That has the villi, again, trying to link your ideas. It's an exchange surface, so it's close to a blood supply, which will quickly take the nutrients away and maintain a concentration gradient, and all the small pieces will go into the blood. The pancreas is hidden behind here. This is where all three enzymes are produced as well. So three enzymes produced in the pancreas, but the food doesn't go into there. It actually goes into the small intestine, which also produces the enzymes. Moving on, defense against disease. The blood contains several things. It contains red blood cells, and they have no nucleus because they just need to bind oxygen. Okay, that's all they need to do. And their red pigment is called hemoglobin. Platelets. Cell fragments help the blood clot. Okay, so the cell fragments in there help the blood to clot. Plasma is the liquid, so throughout the blood you will have liquid, so that will not only carry the cells and the platelets, but if they talk about substances, you're going to want to talk about things that are dissolved in the blood plasma, like hormones. You know, in the menstrual cycle, hormones travel in the blood. Carbon dioxide is dissolved in the plasma as well. White blood cells then. Okay, we've got our red ones, we've got our white ones. The white blood cells fight disease. So think to yourself if you know the 4 things that they can do. So one thing, produce antibodies, and they will match on the antigen of the pathogen. So if this is the pathogen trying to fight your body, it's got antigens on the outside, and your body produces antibodies to match those in a specific shape. Your body also produces, your white blood cells produce antitoxins, because pathogens produce toxins that make you feel ill. So these antitoxins neutralize the toxins. And finally, the reason why I said 4 things is because they can engulf pathogens. And as a 4th mark, you may need to recall the word phagocytosis, which is when the white blood cells will come to a pathogen and engulf it and then digest it. So do not eat. They don't have mouths. Okay, remember they're just the white blood cells. So talk about engulfing. If you want to prevent yourself against disease, we said the hydrochloric acid in the stomach. The skin also produces antimicrobial substances. And you might want to take a vaccine. A vaccine is a dead or weakened form of the pathogen. Okay, so you might want to take those to prevent disease as well. Okay, a couple more things, not too much left now. So we've got cardiovascular disease. If you eat lots of cholesterol, the bad cholesterol is LDL cholesterol. Okay, that's bad. That's not good. LDL cholesterol is bad. And what it will cause is fatty deposits building up in your arteries. Now, the heart itself, as well as all the arteries that come out of it and the veins that go into it, the heart itself has its own supply of arteries that wrap around it. Those are called the coronary arteries. And if these get blocked, it reduces the blood flow to the heart, and therefore, if we try and link our ideas, it will reduce the oxygen going to the heart, and that will affect the respiration and the energy that the heart can release through respiration. So it's going to affect its ability to pump the blood around. So with coronary arteries, you could, if you've got a blockage, you could treat it with a stent. And this is a metal device that requires an operation. You pop it in and it will open out your artery to make it wider and increase the blood flow. Or you can take statins, which are drugs which reduce the cholesterol. On the topic of non-communicable diseases, these are diseases that are not caused by pathogens and diseases that cannot spread. So tumours and cancers come into this group as well. So there's benign tumours. They are cells that will uncontrollably divide but will stay in the same place. And you've got malignant tumours. They will uncontrollably divide, but cells can break off and travel in the blood, and therefore you could end up with tumours elsewhere in the body. And this one is the one that is cancerous. Risk factors then for non-communicable disease, it will depend on the disease, but things like smoking, alcohol, obesity, and having a high fat or a high salt diet.